Pierre Sens from Institut Curie. Pierre Sens runs his group working on theoretical approaches to understand dynamics, principles of organizations of living matter at the scale of single cells to tissues. Endosomal endosomal network or vesicular trafficking is of course complex, and Pierre Sens is probably one of the very few, if not the only one, who has dwelled into this subject with theory. We use many of his papers as our modern new age textbooks for endosomal trafficking and ideas that come out of his papers. So I'm sure that you will enjoy this fantastic session. Pierre, take away, please. Thank you.
Thank you very much, Senthil. Good afternoon, everybody. That was a very nice introduction. So to be honest and precise, I will not really talk about endosomal trafficking per se. I will mostly talk about non-equilibrium models of organelles organization, and I will mostly after having talked about some general concept, I will mostly focus on the case of the Golgi apparatus, actually. So rather the secretory pathway than the endocytic pathway. But hopefully what I will talk about will also have relevance for the endosomal trafficking. So as compared to the first talk of this session from Mahak, there won't be, unfortunately, at this point, a lot of overlap in the sense that I will not really present theory that are able to model her data, mostly because she focuses a lot on spatial organization. And here I will not talk so much about space, unfortunately, simply because I haven't done much on it yet. But it is certainly a desire of me to go in that direction in the future. Okay, here we go. So okay, so this is a very crude introduction. Of course, I don't need to tell you guys about this, but basically all these eukaryotic cells are compartmentalized into organelles. These organelles are drawn on this sketch here are membrane-bound organelles, which means that they are basically surrounded by a very thin, flexible, fluid, and deformable lipid membrane. In recent years, we've heard a lot of interest and discussion on membrane-less organelles, which are basically organelles that are not bounded by a membrane. I believe that many of the concepts I'll talk about are also relevant for these kind of organelles because they are basically a way to describe phase separation in an active system. And of course, the concepts might be the same, the details might be different, in particular about the dynamics, the exchange processes, whether you're bound by a membrane or not. But hopefully the concepts are shared between these two kinds of organelles. And here I'm mostly interested in organelles along the trafficking pathway, so the secretory pathway from the endoplasmic reticulum, let's say, where many proteins are synthesized to the plasma membrane through the Golgi apparatus, or the endocytic pathways where material taken in by the cell through endocytosis is then processed and distributed by endosomes and lysosomes, as we heard earlier today. So the main point here is that when you look at these different routes, these different pathways, what is clear in, for instance, in this sketch, which is a very famous sketch, is that there's a lot of arrows going in all directions, which is supposed to tell you that everything is very dynamical and that many of these organelles exchange with one another. So here we've got the endoplasmic reticulum, which can be seen as a source of, a source of material for let's say the Golgi apparatus, which follows along this pathway. Here we have the plasma membrane, which can be seen as the source of material for the endosomal network and the lysosomes. And of course, there is a lot of interaction between these two modules, the secretory and endocytic modules. And in this sketch, it's the focus of this sketch is on these little protein codes, the blue, red, and yellow protein codes that are in this particular sketch discussed as being the main actors allowing the exchange between the different organelles. So here you have another sketch where you see it might be a bit small, but you see basically that from this donor organelle to this receiver organelles, a material is being exchanged by being packaged into a vesicle itself coated by proteins and then transported either by diffusion or maybe using molecular motors working on tracks such as microtubule tracks, being transported towards the receiving organelles. And here you have a lot of proteins, the snare complex and all these things that are promoting the fusion between the vesicle and the receiving organelles. And so this is one basic step of exchange between different organelles. There are others. You can also have membrane tubules and probably also maybe in some cases simple diffusion. But this, I will mostly discuss this vesicular exchange between the different compartments in my system. Another aspect that is really interesting, which we heard about earlier, is the fact that these organelles themselves undergo maturation, which means that if you look, for instance, at an early endosome, which is characterized by some particular protein and enzyme distribution, you can see that over time, this endosome, which has an identity of an early endosome, whatever identity means, I assume that you can define an identity. Then this endosome will eventually evolves, early endosome will evolve to become a late endosome, which means that basically its protein content has changed. And in this case, it's mostly the composition of its membrane that has changed. Of course, its internal properties have also changed. The pH has changed, and the structure might have changed. But at least from the modeling point of view, I would be interested in introducing the fact that the identity of membrane composition or other can evolve over time in a way that is essentially irreversible. It's quite, I mean, as far as I know, it's not very often discussed that a late endosome can become an early endosome. So we will probably assume that this step is rather irreversible. And the main question I want to discuss is how are those transport pathways defined? How does this vesicle knows that it needs to fuse with this ER and not to go fuse with the early endosomes or the late endosomes? How are these pathways defined in space and time? So in my case, so we heard a lot in the previous talk about spatial structure and organization. And this, I guess, so one possible answer for this, how these pathways are defined is by defining the tracks. If you manage to self-organize tracks, for instance, microtubules, which are polar, and if you manage to organize the polarization properly, then you can say that then the vesicle can just bind on these microtubules, and depending which motor are carrying them, they will go one way or the other. And this is basically, this could be sufficient to define the entire pathway by just defining the tracks. But of course, then you have to answer, how do you define the tracks and how do you specify the location of the tracks? And of course, the microtubule distribution can be quite complicated. And so this is an interesting problem on its own, which I will not discuss at all here. What I'm interested in here is to discuss different kind of models where basically these pathways are organized through specific interactions between the different carriers. So if you imagine different membrane compartments, for instance, very crudely here, something at the center, maybe the nucleus, the endoplasmic reticulum, and something at the periphery, maybe the plasma membrane, and they have different colors, which means that they have different compositions of lipids, of proteins, and various things. Then if these compartments emit some vesicles, which let's say have a similar composition to themselves, and if these vesicles are not directed by any tracks, but just go diffuse around, which is probably quite inefficient, but let's assume that what happens, then these guys will fuse with other compartments in the system according to their colors. And for instance, a vesicle that has a purple color here might fuse easily with a compartment that has a purple color. This is typically called homotypic fusion mechanism. And then within this compartment, you can have maturation. So the purple color can become green color or the other way around. And then the vesicle that will be emitted by this compartment will then be a green color, and this can be directed towards, let's say, the plasma membrane, not by directed transport, but simply by diffusion again and specific fusion. So this is the sort of concepts that I want to explore theoretically. And of course, there could be multiple other mechanisms to organize these pathways, and most certainly a combination of these two mechanisms. So for this second hypothesis to be viable, you need to be able to define identities, and you need to be able to define multiple different kind of identities, because there are multiple different kind of compartments, and so for this you need to have families of proteins or enzymes that can play this role. And although I will not be at all molecular specific in this talk, it's still worthwhile to say that a good candidate for these molecules that would bestow an identity to a compartment is the Rab superfamily of GTPase, the Rab GTPase superfamily, which contains in human a lot of different members, more than 70 different kind of Rabs. And if you look at different compartments, you can typically see different kind of Rabs. So the Rabs are numbered, Rab 1, 2, 3, 5, 7, etc. And for instance, between the early and late endosome, early endosomes are rich in Rab 5, late endosomes are rich in Rab 7. And this is one way, at least, to distinguish them. Other ways are maybe their size and their shape. But if you do fluorescence microscopy and if you image Rab 5 or Rab 7, you can quite readily see whether you're dealing with an early or late endosome. And so basically, when I talk about identity, I will not be specific, but one certainly important contributor to this identity would be what kind of Rab there are on the membrane of these organelles. And these Rabs are interesting because they are known to affect all the possible steps of transport. They are known to affect budding. They are known to be able to select which cargo will go in which vesicle. They're also known to interact with transport molecules such as motors to then affect transport, and to interact with snares to affect fusion. So it is a reasonable hypothesis to say that the exchange between different compartments through the budding transport of fusion could be identity specific through, for instance, the interaction with the Rab molecules. Those Rabs are active molecules. They consume energy, and they have active and inactive state. The inactive state is a cytosolic state where the Rab diffuses around. The active state is a membrane-bound state, and the Rab cycle between the two by hydrolyzing GTP, and this is of course helped by GAPs and GEFs as usual. But so basically, you already see that this is a completely non-equilibrium system because energy is spent to maintain or change the identity of these membranes. And then there is another interesting properties of these Rabs, and probably also other identity markers, which is that there is a so-called Rab cascade, which tells you that some Rabs, so in this case it's the red Rab, is able to inactivate the blue Rab. So if you have a blue compartment and then somehow, for some reason, a red Rab is coming to this compartment, this will tend to deactivate the blue Rab. And so this is a mechanism by which a blue compartment can turn into a red compartment in an irreversible manner. So this is due to the cooperativity of the activation-inactivation cycle of Rabs. But this is one basis by which the maturation of early endosome to late endosome can be explained. I'm sure there are others, but that's one way. So basically, this is, I think, the only molecular specific slide I have, but keep in mind that when I talk about identity, this is what I might be talking about. All right, so now we have these different processes that are at play. We have budding, we have fusion, transport here, and we have maturation. And we can already ask a very conceptual question, which hopefully is of interest for biology, but I think it is, and also definitely for physics, is let's say that you have a system with an influx coming from some source, and an outflux going to some sink. And let's say that the source is early or white or immature, and that the sink is late or mature or black in this case. And you can imagine two extreme way of self-organization of the system in between the source, the source and the exit. One possibility is that you will form stationary compartments in between those two, and that the vesicles that are emitted by the source will fuse with this compartment. In this compartment, due to the enzymes that reside in this compartment, they will undergo some sort of maturation from early to late identity. And then when they are late, when they are mature enough, they will be emitted by this compartment and they will go fuse with the sink. And then in this case, you have a steady state. You have a real steady state in the sense that this compartment always exists. Of course, the molecule that compose this compartment vary over time, but the identity of this compartment exists, and basically this compartment can be used as a processing factory to process early component into late component and then emit them. That's one possibility. Then another possibility is that these early identity vesicles that are emitted by the source will sort of fuse together to form a compartment, but at the same time this compartment undergoes maturation, and eventually the compartment will have an identity that is so different from the source that fusion will not be possible, and so this compartment will effectively become isolated from the source. It will go on maturing, becoming a late identity, and then this entire compartment or vesicle emitted by this compartment will fuse with the exit. And in this case, you don't have a steady state because the compartment evolves over time and eventually disappears, and a new compartment forms, evolves over time and eventually disappears. So at the very broad sense of fluxes, you suddenly have a directed flux, and you have a steady state, but with very strong fluctuations in the number, size of the compartments that reside in the system at any given time. And so basically what I want to do is to study models that can discuss which mechanism or which self-organization is more likely to take place depending on the kinetics of the different processes involved. And I will want to discuss the Golgi apparatus in particular because I'm fascinated by this organelle. So this is an example of an electron tomography of the Golgi apparatus of a plant, of an algae, I guess, in this case. And this is another EM picture. And what is fascinating here is, in this case, the spatial organization, which is really incredible. So basically everything that you see here is a membrane, so a very thin 5 nanometer thick lipid bilayer. And all these structures that you see are basically vesicles. They could be very small vesicles like these white dots here, which are the vesicle, the sort of 50 nanometer diameter vesicles that do the transport between the different organelles. There could be larger vesicles like these guys, which are very, very flat. They are called cisternae for the Golgi. And in this particular case, you can see that the cisternae are stacked on top of one another. The size of this whole picture is about 1 micrometer. So you can see that this organization is very small indeed at the scale of the cell. And here also you have this 1 micrometer scale bar here. And what is also amazing is that although this structure seems fairly well defined, it is completely out of equilibrium. That's obvious. And it is traversed by fluxes all the time. So if you look at the composition, if you look at one lipid molecule that would be here at a given time, later it will be there and eventually will go away. So this whole thing is traversed by fluxes. Here you see in yellow the ER, you can see the ER here. So this is the source of material for the Golgi. And then so the flux basically goes, at least the flux of protein synthesized in the ER goes through the Golgi. It enters through the phase of the Golgi, which is called the cis phase. And then it goes through the medial part of the Golgi apparatus, which is called medial Golgi, and then it exits through the trans phase of the Golgi stack. And in the process of its transport, it is being processed by enzymes proteins are being glycosylated, or a lot of processing is happening there. Of course, with this processing, a lot of proofreading is also happening there. So basically, this system here is a processing factory for the proteins secreted by the cell. And so this structure is quite amazing. Unfortunately, today I will not talk about spatial structure very much, as I said, but still what is also amazing is this organization. So you've got different compartments that are well defined. They have a well-defined identity. So these cisternae here are of cis identity. These one are of medial identity, and we don't really see the trans identity here, but they are at the trans phase. And this is the case for mammalian cells, for plant cells, for algae. But if you look at different systems, for instance, yeast here in Cerevisiae, the Golgi doesn't look at all the same. It actually, all these different cisternae that are dispersed in the cell, and so the organization is completely different. So the fact that they are dispersed is nice in a way, because it means that you can follow individual cisternae. All these different dots with different colors here are individual cisternae, and you can follow them over time, and you can do fluorescence microscopy to follow their identity. So the red ones are the cis identity in this case, and the green ones are the trans identity. And if you follow one cisternae over time, you can see that the cis identity goes down and the trans identity goes up. And so this is basically showing that just like the endosomes, which goes from early to late endosome, these Golgi cisternae in yeast, at least, they go from cis identity to trans identity. And so it looks like these two different systems here sort of illustrate these two different possibilities that I was telling you about before. And so the same organelle apparently can behave in both ways in different species, although many of the proteins are the same or have equivalent between the species. Of course, there are differences. And one interesting difference, which might not be a cause, it might be a consequence, but it's still quite interesting, is that if you look at the typical time scale of cisternae maturation in yeast, it's about 1 minute, as you can see here in this time axis. While if you look at the typical time scale of transport through the Golgi of mammalian cells, it's about 20 minutes. So we already have 1 order of magnitude in typical timescales between these, between the dynamics of the Golgi apparatus in these two different species, and we have a very different structure. So of course, it begs the question: are the two things related? Is the fact that the structure is very different due to the fact that the dynamics is very different? And this is basically the subject of what I want to tell you about today. Right. Another thing that is interesting, so here this is in the case of endosomes. So if you look at an early endosome and you follow it over time, which you can do, and then you can look at the size of the endosome. And this is one example. I'm not saying that they all behave this way, but this is one example from this publication here. And you can see that the size, it has a lot of fluctuations, but it reaches some sort of steady state size for a while. And this is also in seconds. So you have a steady state size for something like 500 seconds, and with fluctuations, of course. If you look at the identity, so this is the Rab5 marker. So this is the amount of Rab5 fluorescence there are in this early endosome. You can see that there are really, really large fluctuations. Obviously here, the Rab5 identity just jumped a lot, which means that probably another early endosome just fused with that endosome. You can see it goes with the jumps of size as well. And then the early identity, the Rab5 identity, goes down with time, and then it jumps, it goes down, it jumps, it goes down, etc., etc. Eventually, at some point, it keeps going down, and it goes down to 0 or something close to 0, which means that this organelle has just undergone maturation to a late endosome. So this is my picture, at least, of early endosome maturation. Is a random walk, basically, in a composition space where you've got jumps and you've got decay and jumps and decay, and eventually you have a full decay to a late endosome. And for those who like stochastic phenomena, this is very reminiscent of a random walk with an absorbing boundary condition here, once you become, once the early identity has disappeared, then you are committed to being late endosome. And of course, so the maturation time in this case is the time it takes for this endosome to become a late endosome. And there are strong variation. If you look at different early endosome, some of them are doing really, really fast. Others are doing it slowly, and even with something that almost look like a cyclic maturation and a cyclic variation of the early identity. So the goal of this slide is to tell you that everything in this system is very stochastic. Okay, so what I will tell you about is to describe this sort of process by using models that are basically models of fusion, aggregation, and scission, controlled by stochastic equations, or the mean field version, which is basically what I will discuss. And this is one way to deal with a phase separation phenomena. And why do I want to call this a phase separation phenomena? Maybe this will become clear in the future, or in the subsequent slides. And this is basically what the theory will look like. So this is this equation here tells you how the number, so basically we have a bunch of compartments. At this point, there is only a single identity, which is it's basically there's a bunch of compartments. This compartment can have different size. N is the size of the compartment, and small n and capital N is the number of these compartments that exist in the system at a given time. You can also use composition instead of number if you want to take an infinite system. And you can write a differential equation for how the number of compartments that have a given size evolves with time. And in this particular case, we only consider fusion. So we say that we start with a bunch of different compartments and they can fuse with one another. And they fuse in a stochastic way with a fusion rate, which is called Kf here, which is constant in this case, but it could also be size dependent. And so this equation, which is called the Smoluchowski coagulation equation, tells you that if you want to know how the number of compartments of a particle size evolve in time, it evolves in time because if two smaller compartments of appropriate size fuse together, they can form a compartment of the size you're looking at. And so this is a positive term, which tells you that the number of compartments of size n will increase if a compartment of size m fuses with a compartment of size n minus m. And this occurs with a given rate. And you have to sum over all the possible m, which could give you the right size. And then this number decreases over time if the compartment of size n that you're looking at fuses with another one to form a bigger compartment. And so in this case, you obviously have to sum over all the other size, the size of the other compartment that are possible. And so if you have this equation, then you can solve this equation. So either you can solve it analytically, if you're lucky, if in particular, if these rates are constant, then you can. Or if the rates are not constant, maybe because fusion is limited by diffusion of this compartment, and that the diffusion depends on the size, as it would in simple 3D diffusion. Then you cannot solve it analytically, but you can solve it numerically. And typically what you have size distribution evolve in time. So in blue, you have the early time size distribution where a lot of the compartments are small and very small, very few compartments are big. And over time, this distribution evolves like this. So for the number of small compartments decrease and the number of large compartments increases. And if the rates are constant, then you can find some sort of asymptotic analytical expression for how the number of compartments of size n evolve in time. And you can see that you have an exponential decay with a characteristic size that itself evolves linearly in time in this particular case. So basically, what you can say is that you will have a lot of small compartments, very few large compartments, an exponential decay with a characteristic size that evolves linearly in time and depends on the fusion rate and on the total mass of the system, which is basically how much mass you have, which is defined as the sum of, there's an n missing here, sorry. So, and you can also do computer simulation. This is an example of a movie where you start with only single, like let's say small vesicles, and then you let the vesicle aggregate, and you see that eventually you get more and more mass into the bigger compartment. And if you wait long enough, you'll end up with a single large compartment. Right. So, it's okay. Maybe I just pause for a second and see if we have questions on this. If that's a good idea.
Yes, thank you, Pierre. Vijay, do you want to go ahead and ask a question?
Yeah. So Pierre, this was about the cisternet that you showed in the east. You said they're not stacked like in eukaryotes or other kinds of cells. They're sort of mobile, moving around. So if they're mobile, I mean, I don't know, if they're mobile, then is there a well-defined current of vesicles across the cisternae? If they are stacks, then there is a nice...
So unfortunately, these vesicles are very small, so they are about 50 nanometer, and maybe it will change due to the improvement of microscopy or live microscopy, but there's no, as far as I know, there's no measurements of how the vesicles are being exchanged between these compartments. And this is, of course, one big question, and if we knew that, that would basically simplify a lot our understanding of the process, but I think it is not known yet. But I will discuss vesicular transport between compartments later, but mostly from the theoretical point of view.
Okay.
Srilaja?
Yes, my question was more to do with the size evolution. So the inherent presumption here is that larger sized vesicle come from smaller sized vesicles. But it is also
about the model, or are you talking about the real life?
Both. I mean, in theory, the model you showed. So, I mean, in real life, perhaps de novo certain sized vesicles emerge, and it is also possible that fusion and fission generate smaller or larger vesicles.
Yes, this will come. This will come.
This
is just the first step.
Maybe
so I will, of course, in this particular model, there's only fusion possible. So the evolution is irreversible towards larger size, but then you can have scission, you can have vesicle budding and all this. This will come in the subsequent slides.
Okay, so then I will wait.
Abhishek, could you unmute yourself and go ahead?
Hello, sir. I was just wondering about the second part of this equation, because I did not understand. First part also involves correlation and increasing in the size of the compartment. The second also has the same, but I don't know if I probably might have missed it. I don't know why the second term in this equation is present. Could you please?
Right. So this equation does not tell you about size. Well, it tells you about the number or the concentration of compartments of a given size. Okay. So then if you want to know of course, there is no single size in the system. You have compartment of many different size, and you can expect an almost continuous distribution of compartment size. So this is the size distribution, right? This is the number of compartments that have a given size, and this is the size here, small n. And basically, what this is saying is that if you look at one compartment which has a size n, and you want to know how many of those compartments there are, you can ask, how do you create a compartment of size n? And in this equation, in order to create a compartment of size n, you need two smaller compartments to fuse together. And then you create one, and this is why you have a plus in front of this first term. But then you also want to know, how do compartments of size n disappear? And they disappear by fusing with another compartment of any size, and then they make a bigger compartment. So this compartment of size n has disappeared in the sense that it has merged with another one to become a larger compartment, hence the negative sign here.
Okay, thank you.
You're welcome. Okay?
Yes, Pierre, you can go ahead. No more questions.
All right. Okay, so so this is one way of describing phase separation, because basically at the end of the day, we end up with one large vesicle, although or compartment, whatever it is. But initially we started with a lot of small compartment. So in a way, you could describe this certainly as a phase separation. I mean, if those were, let's say, water molecule in the gas form, and then this would be a water drop, then this would be a phase separation, right? So of course, phase separation generally can be described in many different ways. It can be described as a continuous process with a continuous field, which is often the way it is taught in school. So this formalism can be used also to describe phase separation. And I will just want to show you some example of this. So I draw the example from membrane phase separation in multi-component membranes, because this is what I had in hand. And so this is a giant vesicles, which is made of two kinds of lipids and a cholesterol, and this typically leads to phase separation. You can have a phase separation between different phases. One phase with mostly one lipid, and the other phase with mostly the other lipid and cholesterol. And as you can see in these fluorescence images, and then you can have phase diagrams. This phase diagram in this particular case would be ternary phase diagram, because you have three components, and depending on the temperature. If the temperature is very high, then you will not have phase separation. You will have a mixture. If the temperature is very low, then you will have phase separation. And at a given temperature, then you have phase separation inside this gray region, which corresponds to a mixture, which is basically where all the components are present. And then you can, so this is a movie or the phase separation happening at the surface of a lipid vesicle. This is a movie, an old movie by Sarah Keller. And what you can see is you've got these different kind of domains. They diffuse around, they coalesce. I think these two are going to coalesce at some point. And so this, here you are. And so this looks very much like the equation that I told you about, right? And so domains have different size, but you can define a characteristic size, and then you expect this characteristic size to increase with time with a particular power. So this is, you expect a dynamical scaling law, basically. And depending on the process through which these guys will exchange material, you can have different dynamical scaling law. But at the end of the day, if you wait long enough, you will have a macrophase separation, which means that you will basically form one big domain. This is what thermal equilibrium will tell you most of the time. Thermal equilibrium will tell you that either you have a dispersed phase or you have a full phase separation. You can have long-range interactions that prevent full phase separation to occur, but probably not in biological systems. So, and then the dynamics could be different, so I won't get into the details, but basically in the dynamics that we saw in the movie, you've got diffusion, coalescence, and then you expect this exponential size distribution, and then you expect the scaling law to be that the size is like t to the power 1/2, but that's not really well, it's important and interesting, but you can check that experimentally. Now, if you look at dead systems like these, you end up with a full phase separation. If you look at living systems, so this is a yeast cell again, and the colors here represent the different compositions in proteins and lipids of different patches in the cell membrane. And as you can see, this looks nothing like that. And there could be many reasons. First, there could be the fact that there are many components, while here we only have 3 and 2 phases. But another obvious reason is that this is not at equilibrium. You do not expect this system to behave as an equilibrium system. And actually, out of equilibrium processes can both promote phase separation and prevent full phase separation. And so different non-equilibrium process can be thought of. For instance, for membrane, the fact that there are proteins that cycles between active and inactive cycles, as I said, or the fact that you have endocytosis and for the plasma membrane, endocytosis and exocytosis that basically mix up the composition of the membrane. Even if this compartment has a steady state composition, the fact that you bring things in and take things out all the time will be a bit like if you had a glass of water with oil in it and you expect to have a big oil drop in it, but if you stir your glass of water continuously, then you will break up that big oil drop. And this is a bit what this is doing. Another possibility is that you have a cyclic chemical reaction which turns the dark red into a light red. And if the reaction rate is small, then you can have large aggregates. If the reaction rate is fast, then you have smaller aggregates. And so basically it's interesting to think that if you have this equilibrium phase diagram, but you have a non-equilibrium process, you can imagine that at steady state, you might be outside the phase separation region, but that activity could promote phase separation, or you could be inside and activity could prevent full phase separation. You could also be exactly at the boundary, which is an interesting possibility. So examples where activity could promote phase separation are examples. So this is the work by people in Bangalore, actually Madan Rao, who's going to speak tomorrow, where they looked at proteins that were carried by on actin in this case, but it could also be microtubules. So these kind of models where you have stuff that are carried by, let's say, on microtubules toward the region where they are aggregated by active processes, and then they could coalesce, and then so this could basically drive a high concentration in a particular location and lead to phase separation. And this could be an interesting model to study the distribution for endosomes or lysosomes that we heard about before. Processes where activity could break phase separation is, as I said, chemical reaction, or the fact that you recycle material. And then some people argue that plasma membrane might be exactly at the boundary and behave as critical systems. I don't want to get into that, but this is one possibility. Okay, so this is the end of the digression I want to make. And now I'm going to introduce budding in my model. So let's say that I have a system where I have different vesicles or compartments that can fuse with one another, but I also have a process that can break up my compartment. That this could be scission, so the compartment could break into two compartments of smaller size, or this could be budding, where basically the compartments that are being emitted are always the same size, which is the size of a vesicle. But if you imagine that the fusion and the budding depends on the size of the system in different ways, then this means that there could be a particular size for which the fusion rate and the budding rate would balance each other, and you could have a steady state. So in this case, you could imagine having a steady state for a particular size, because at this size, the flux of growth coming from fusion would balance the flux of shrinkage coming from budding. And then you have to modify your Smoluchowski coagulation equation. So you still have the fusion terms, which are the same as before, but then you also have to break up. So here in this case, I have a budding phenomenon, and I assume that I bud compartments which have a size 1 being the size of my vesicle. So in this case, I will increase the number of compartments of size n if I have a compartment of size n plus 1, and this one undergoes budding. So this will then become a compartment of size n. And it makes sense to think that the budding rate should be proportional to the size of the compartment, because if I have a bigger compartment, I can have more vesicle coat forming and I will have more budding. So this is what this term here is saying. You've got a budding rate times the size of the compartments times the number of compartments of size n plus 1. And then, of course, if a compartment of size n buds, then it becomes a compartment of size n minus 1. So you've lost it, and so there is a minus sign with the same kind of term. And if you want to look at the number of vesicles you have in your system, so vesicles are compartments of size 1, then each time you have a budding event, you add one vesicle, which is here, the number. So basically all the vesicles that have budded from any compartment of any size, they become a vesicle. And so this is a term that is peculiar for the vesicles. And so in a very crude way, if we think that we have a stationary size distribution, since we know that without budding, we know that the size of the aggregates evolve linearly in time, then with budding, the stationary size distribution could simply be given by this expression where I replace the time by the dynamical process that gives me the stationary state, which is the budding rate. So in this case, I would expect a characteristic size to be equal to the total size of the system times the fusion rate divided by the budding rate. So here time has been replaced by 1 over the budding rate. And indeed, this is what you find. So the typical stationary size distribution in this case, which means that although fusion and budding are happening all the time in this system, I still have a stationary size distribution. What I observe is that I have a power law at small size, and I have an exponential cutoff at a particular size, which happens to be the size that I told you about before. So I will have a lot of small vesicles, I will have very large few vesicles, and the crossover between the two regimes is basically a size that depends on this rate. If the fusion is fast, the size gets larger. If the budding is fast, the size gets smaller. And there could be a particular situation also where, since I have a limited number of things in my system, this is a finite system, if the fusion rate is larger than the budding rate in this particular case, I still end up with macrophase separation, which means that I still end up with one big compartment. Okay, so in this case, the main control parameter is the fusion rate over the budding rate. And this is what will control the state of the system. Okay, so so far I only told you about a single component. But of course, in cells, there are multiple components. This is a picture that is showing you the distribution of lipids in the cell. So which lipids you find in the plasma membrane, which lipids you find in the plasmic reticulum, etc., etc. And as you can see, the distribution of lipids is very different in the different part of the system, although these guys are exchanging all the time. So basically when you exchange things, this should lead to an homogeneous distribution. This is basically how diffusion works. Diffusion leads to an homogeneous distribution. And the fact that the distribution is not homogeneous means that you have to actively sort lipids, and you have to actively remove from the plasma membrane lipids that are not very much present there, and actively move them to other places where they would be very present. And same thing with all the other organelles. So how would sorting work? Well, one way that sorting could work is if you have a mixed system and you specifically export yellow and red components by vesicles. So let's say that you bud vesicles that only have yellow, you bud vesicles that only have red. And then to this, you add the fact that, let's say you want homotypic fusion. So you want the yellow vesicle to fuse with yellow compartment, but not to fuse with red compartment. So if you have these two ingredients, you can expect that eventually this system of a mixed vesicle or mixed compartment will separate into two independent systems with only yellow and only red. Of course, this separation will be slowed down by the fact that when you emit a yellow vesicle, this yellow vesicle can fuse back with your compartment if there is still yellow identity in this compartment. And so one interesting question is, how much time does it take to separate yellow from red by this process here? And you can study this by basically having the same sort of model than before, except that now you have two colors, A and B, and the way fusion occurs depends on the how similar is the composition of this compartment with the composition of this vesicle. And here phi is the fraction of B or red that you have in this compartment. And of course, you need to say that if you want homotypic fusion, you need to say that the fusion rate increases if the composition of this compartment is the same than the composition of the vesicle, which you can use different kind of mathematical form for this. Here I have a mathematical form which looks like a Michaelis-Menten kinetics, but it doesn't matter. And this is for the budding. Sorry, so the fusion looks like this. So the fusion basically is simply saying that if the composition of if the composition of this is the same than the composition of that, then the fusion will be high. And then you can simulate this system. And this is what happens if you start with a compartment that has a mix of A and B, so phi is equal to 1/2, and you only let B being secreted. So if you let B being secreted, it means that phi will go down, but eventually the B's that were secreted, they can fuse back. So boom, phi goes up, and phi goes down, up. Budding, 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 fusion, etc., etc. And eventually, if you wait long enough, you will end up with a system where all the B's have been exported, and you have a compartment which is full of A. And you can study how long this will take. And I will not get into the details of the mathematics because I'm actually taking a lot of time speaking. But basically, what you can see is that there's two different kind of limits. One limit is when fusion is very slow. So in this case, the time you need to completely expel all the bees is basically the budding time, because sorting occurs through budding. So if the budding is much faster than fusion, the time you need to completely sort the system is proportional to the inverse of the budding rate. On the other hand, if the fusion is fast, then each time you export a bee, it comes back. And so you can expect that the sorting mechanism will be very, very slow. And indeed it is exponentially slow as a function. It's slow with the exponential of the ratio of the fusion over the budding rate. So you have a transition between a very fast sorting and a very slow, exponentially slow sorting. This is in log scale, when you vary the ratio of fusion over budding rate. And so now, so this tells you that the sorting kinetics very much depend on the ratio of the budding over the fusion rate. So now, if you couple this with the fact that the size distribution of the compartment once they are sorted can also depends on the ratio of the budding over the fusion rate, and that if fusion is very fast, then you have large compartment, but if fusion is very small, then you have very small compartments. You can have a sort of sorting phase diagram, which looks like this. So here you have the ratio of budding over fusion. Here you have the composition of the initial compartment. And what this tells you is that if you want fast sorting, you need the budding to be much faster than fusion. But then if you have that, you will end up with a very, very small vesicle, with a distribution of very, very small vesicles. If you want to make big compartments, then you need fusion to be faster than budding. So you're here, but then in this case, you will have very, very slow sorting. And if you want to, at the same time, being able to sort efficiently something, and to also end up with large organelles or large compartments, then you need to sit somewhere in the middle, and it's a delicate fine-tuning of parameter for this to happen. Okay, so this is to illustrate through the sorting, vesicular sorting mechanism, that basically rates are extremely, I mean, the stationary state that you get is extremely sensitive to rates, and also the kinetics at which process occurs. We see here an exponential dependence of the sorting time as a function of the ratio of budding over fusion rate, which is a very, very strong dependence. And then, so here, this is just to tell you how can we use this to maybe explain some data. So here, these are data where using a drug treatment, BFA, which BFA is a drug that causes many things, but in particular, it causes the Golgi apparatus to completely fuse with the endoplasmic reticulum. So if you use BFA, you lose the Golgi apparatus. It goes to the endoplasmic reticulum. And then when you wash out BFA, you can ask how much time you need to wait for the Golgi apparatus to appear again. And of course, in this case, if you look at this system with my very crude model, A would be components of the endoplasmic reticulum, to be very crude, and B would be component of the Golgi. And I use BFA, so I killed so basically I completely fused the Golgi with the endoplasmic reticulum, and then I wash out. So Golgi material can be exported for the endoplasmic reticulum, but it will come back because there's still a lot of Golgi material in the ER. And you need to wait long enough for enough Golgi material to be exported to form a new Golgi. And this is what it looks like. And you see that the time you need to wait is about 20 to 30 minutes, which interestingly is the same kind of time than the transport time through the Golgi apparatus. And then we sort of try to estimate what would be the budding rate and what would be the fusion rate in this case, which is very hard to do because, I mean, it's not easy to know what these parameters are like. But what we concluded is that this process is a slow process, which is basically consistent with the assumption that you are in the slow sorting regime and that you are somewhere around here. You're not very, very slow, but you are slow. Basically, the Golgi reformation time is about 1 order of magnitude larger than it would if there was no fusion back from the between the Golgi and the ER. So using this model, what we say is that this kinetics is consistent with the fact that Golgi reformation after drug treatment is slowed down by the fact that Golgi fuses back with the ER as it is exported. All right, so I think I will pause at this point because I'm going to change the subject a bit, and I would like to ask if there are some questions. Hello? Are you guys hearing me?
Yes, okay. Yes, we are here. Amir Sohail?
Yep. Hello?
Yes, hello.
Hi.
Hi, Amir. You can go ahead and ask your question, please.
Well, I was asking now it is clear in later part of the talk. So
Right.
I was asking, does these rates need to be same? Like when you were only considering the fusion, so there was two terms. So I misunderstood something, but it's...
All right, no worries. Sujit Kumar?
Yeah, hi. Can you hear me?
Yes.
Yeah. So I was asking, how do you decide the maximum value of n, which is the size of the vesicles, and how do you discretize? Because your set of equations is a discretized version of compartmental kind of equation. So you have a coupled set of n number of equations, for example. So how do you discretize them according to the sizes? That was my question.
Good, thank you. So in this particular case, this is of course a simplified model. You can make different kind of models. You can make as general a model as you want, but you will always need to say that there is something that is the smallest thing in your system. In this very simple model, the smallest thing in my system is a vesicle. And let's say this would be a vesicle that would be, okay, sorry. Oops. So let's say that the smallest thing in my system would be one of these transport vesicle here, which are protein coated vesicle. Doesn't need to be, but let's say that this is what I want it to be. So these vesicles are about 50 nanometer in size, and these compartments are about 1 micrometer in size. So there is a large difference. So basically one compartment is made of the fusion of many, many, many vesicles. And so in this case, my smallest size, which I called 1 here, is the size of a vesicle. But you always have something that is the smallest thing in your system. I mean, it could be a lipid molecule, but I mean, that would be too small for my purpose. Now, the other question...
Yeah,
go ahead.
Yeah, please. Yeah, so then my next question is, like, when you fix your smallest size, how do you go to the next smallest size? I mean, how much bigger is the next one?
Right, okay. So in this case, if the smallest thing you can have is a vesicle, then the next smallest things is when two vesicles have fused together to form a compartment, which is made of two vesicles.
I see, okay.
And of course, I mean, I guess that if you completely wash out the Golgi, you never maybe completely wash it out. Maybe there are things that remain. Maybe there are remnants that are still attached to the cytoskeleton or something like this that could be used as a kernel or nucleus to initiate the reformation of the Golgi. But actually, the fact that it takes so much time maybe argues that you don't have that sort of remainance that could be used to nucleate the formation of the new Golgi.
Yeah.
And then I think at some point you also had a question about the maximum size. So just in this case, the maximum size is either infinity if you want it to be, or it's simply if you have a finite size system, then it means that you have a certain amount of material in your system, and the maximum size would be when all this material is into a single compartment.
But if you have the maximum size infinity, then you will have infinite number of equations, right, in this set?
That's right. You can have infinite number of equations. Of course, the good thing is that these equations, all these equations at this point, they all conserve mass. You're not creating anything. So you still have a conservation that if you integrate, and this was here somewhere, where was here? Yes, here. Yeah, here it is. Sorry. Okay. So if you integrate the total amount of material that you have in your system, you get a constant. And so this will tell you that beyond a given size, you will not have any
number will be 0. Yeah. Okay, I understood. Thank you very much. Yeah.
All right, Vijay, do you want to go ahead and ask the
Yeah.
question?
So Pierre, in the phase diagram that you showed in the cell sorting, the one that you showed, if you can go to that slide, here. So the symmetry that you find in that red dot, about
Yes.
the phi naught, is that an accident or is that some tuning?
Okay, so this is this is a very simple model. Okay, so basically, what I described here is the fact that you only is what happens when you only remove B. If you remove B and you form basically a distribution of B vesicles that can then fuse with one another, then all the A's that you have, they remain in this compartment, and then you get this line here, the blue line here. But if you can remove B and A, then of course you will have a symmetric system, and then you get the red dots here.
Okay.
So it just means that everything is symmetric with respect to phi equal 1/2, which is when you have as many B's as A's.
Yeah. Okay.
Shailaja?
Yeah, so my question is to do with a loop these kinds of sortings can create. So what is because two vesicles could fuse, make a larger vesicle, and that large vesicle, from that large vesicle, things could bud off. So
Yes.
essentially you have events that are consequent on each other. So, and that might, I don't know if that is accounted for in the modeling that is done, that you can have these events not occurring just independent of each other, but sort of feeding into each other as a feed forward loop, like an infinite series of feed forward loops. So how does that change the outcome of such a process?
So this is definitely accounted for, and actually, when you look at these equations, these are dynamical equations, right? They describe how the composition or number of compartments of a given size evolve in time, and as you can see, this number of compartments at a given time will change over time according to the number of compartments of any other size in the system at that point. And so everything is connected. And what the sort of, so I would not necessarily call that a feed forward loop or feedback loop, or it is just the fact that everything is correlated with everything in the system.
Okay.
And when,
so,
okay. Just finish. And when you have a steady state, so a steady state basically means that this part here is equal to 0. It means that nothing changes over time. And then this sort of distribution that I described, they remain constant over time. But that doesn't mean that nothing happens in the system. Of course, everything always happens in the system. This large vesicle will bud this large compartment will bud more vesicles, and these small vesicles will still fuse with one another to form larger compartments. It's just that all these events will balance each other, so that when you average over all the events, the distribution remains stationary in time. But there's still a lot of activity going on, although the system is stationary.
And what about vesicles that leave the system? So
right. So at this point, there is no vesicle that leaves the system. Everything is closed here.
Okay.
But now I can move to some irreversible phenomena, if there are no more questions.
Thank you.
Pierre, there's one question from Priyant, if you can answer that, and then we can move on.
Yes.
Thank you, sir. I'm curious to know why there's a distinct, distinct, what is it? Distinct difference between the slow fusion and high fusion mathematically. Like in case of slow fusion, you have an inverse relationship, but in the case of high fusion, you have an exponential relationship. I would like to know what is the reason behind such a distinct difference between these two kinds of fusion mathematically. Like why one follows an inverse relationship and the other follows an exponential relationship?
Okay, so I mean, mathematically, there's no difference in the sense that it is not an abrupt transition. As you can see from the numerics, you can see that the numerics go continuously from the fast sorting, where basically sorting is independent of the fusion rate, because fusion is basically irrelevant because it's too slow, and the exponentially slow sorting, where you've got this exponential sorting behavior, but it's a continuous process, right? On the other hand, you can still define a sort of boundary, which is not a real boundary from you know, it's not a critical behavior from the mathematical point of view, but you can also study the system not from a stochastic point of view, but from a mean field point of view, where I wonder if I have that somewhere, maybe here. Yeah, so you can also write mean field equations. So basically, this is a mean field equation that looks at how many B components you have in this system, in this compartment. Not looking at stochastic, not looking at fluctuation, but just looking at mean field. So neglecting correlations that we just discussed before. And then in this case, basically, you can say that, so you can write this equation. So basically, this is the B components, they decrease because of budding and they increase because of fusion, and the fusion depend on the composition. And so you get non-linear equations. And then you can solve this and you can ask, do you have a stationary state? And do you have a stationary state where the system still has B component in it? And when you answer that question, you basically say that, yes, you have a stationary state if this condition here is satisfied. This is a mathematical condition on the ratio of fusion over budding. And when you plot that, you get exactly this line. So basically, intuitively speaking, the crossover between fast sorting and slow sorting is when the fusion rate is large enough so that there is always B component in your compartment.
Okay, sir. Okay, I got it clear now. Thank you so much.
Yeah, this is a mean field description, which of course there will always be a time where all the B components are gone because of stochastic fluctuation, but this time becomes exponentially long if you are beyond that limit.
Okay, sir. Thank you.
Good. All right. So
All right, Pierre, we can move on.
Sorry?
Yeah, we can move on.
Okay, so I see that time is flying. So I think I will go very quickly on this maturation business because I want to get to the end, which is maybe, I mean, maybe the most relevant to biology. So as I said, if you look at one Golgi cisterna in yeast, you can see that it undergoes maturation. So there is some irreversible biochemical reaction process, which could be complicated. It could be due to this activation-inactivation cycle of RABs or other things. Basically, the bottom line is that the green components irreversibly turn into a red component. This is the way I want to model it. And so we also looked at this. We also did model with this, but I think I will just skip this. But maybe I'll just tell you quickly the model. So the model here is that I have a compartment and I have a source, and the source is a source of A components, so early components, and blue components. But when those blue components are in the compartments, they are irreversibly turned into red components. Let's say there's an enzyme in this compartment that can turn this blue into red, and then the red exits by budding. And so I have a process which is influx and fusion, maturation and budding. And in this case, it's an open system. So vesicles enter and leave the system, right? If I do a numerical simulation of this and I look at the total size of my compartment, the total size, it does stuff like this. There is a steady state size. There's a lot of fluctuations. If I look at the amount of A component I have in my compartment, it does something like this. It grows because of fusion from the source, and then it decays because of maturation and budding, and then it grows, it decays, it grows, it decays, etc. And at some point, boom, it goes to zero. And which means that when it goes to zero, if I have homotypic fusion, the influx is suppressed because influx requires that there's some A component here. And so here I basically have witnessed an irreversible maturation of my compartment from an early blue identity to a late red identity. And I can compare this to the endosome data that I showed you, and you can see some similarities. You can also see some differences. And so basically, we studied this, but I'm afraid I will not have time to discuss this very much, which is a bit unfortunate, because I want to move into modeling the Golgi apparatus now in a maybe a bit more realistic fashion. So so the Golgi apparatus, basically, it's here, and it's very complicated, of course. You've got the endoplasmic reticulum, you've got microtubules, you've got the plasma membrane, you have a lot of different kind of proteins, etc., and many different kind of models to understand how the Golgi apparatus works. I won't describe these models very much because I have little time, but I want to tell you about our models. So basically, most of the models that exist, they are either cartoons, which means that they are helpful to understand, but they are not quantitative, or they are quantitative, but then they have many parameters. And by many, I mean about 1 to 500 parameters to be fitted. And so for me, this is maybe interesting, but certainly not helpful in a conceptual level. So I wanted to make a very simple model with as little parameter as possible, with a source. Let's say this is the endoplasmic reticulum, a sink. Let's say this is a trans-Golgi network or something. 3 different kind of identity, because I have the cis, the medial, and the trans identity. I've got different kind of kinetic processes that can happen. Budding, which is specific. So when you form a vesicle, it's of one of these identities. Fusion, which is homotypic. So blue fuse with blue, but blue doesn't fuse with yellow or red. And then maturation, because we saw that there is maturation, where blue can turn into yellow and yellow can turn into green. And I wanted to study this model. And I wanted to use the fact that we have already understood a bit about vesicular-based sorting, and we've understood that if the budding rate is faster than the fusion rate, then the thing will be sorted into compartments of different composition, but if not, then it won't. And the maturation, which I didn't have time to tell you about very much, but basically the maturation shows that depending on the ratio of maturation rate of a budding rate, if the maturation rate is very much faster than the budding rate, then a compartment will undergo maturation quickly and will be isolated from the source, which is giving it material. On the other hand, if the budding rate is much faster than the maturation rate, then whenever you have a compartment that undergoes maturation, then it can be budded very fast, and the system itself becomes in a weakly mature state. So this is a very important parameter to tell you whether you will be in a steady state with a compartment that remains there for a long time, or whether you will have compartments that evolve over time. Okay, so basically the model is this. You make a computer simulation with all these different steps. You need to give yourself some rules for fusion and budding, and that I won't get into the details. What I want to tell you is I will show you computer simulations, and what you will see is a bunch of balls of different colors. The color of the ball tells you about the composition of the compartments. If it's purely blue, it means that it's early. If it's purely red, it means that it's medial. If it's purely green, it means that it's a trans or late. And if it's somewhere in the middle, like a grayish something, it means that it's a mixed compartment with many different species in it. The size of the ball will tell you the size of the compartment, how many vesicles have fused together to make that compartment. And let's start with one simulations where the budding rate is very small. If the budding rate is very small, it means that first of all, you have big compartments, because what breaks this compartment up is the fact that you can bud material. If the budding rate is very small, it also means that you're not able to sort the different components, because sorting occurs by budding different components. And so, as you can see, you end up with large balls which are of fairly undefined colors, which means that they are basically a mix of the 3 different kind of identity we have in the system. Now, if we increase the budding rate, of course, the compartment becomes smaller because they decrease in size due to budding. But the nice thing is that they become much more well-defined in terms of their color. There are basically now there are some compartments that are still big, but they are purely blue or purely green, which means that by increasing the budding rate, you've managed to make the compartments of a composition that is much more specific than before. And if you increase the budding rate even more, then basically you end up with a distribution of vesicles, because as soon as vesicles fuse, they bud and you break them up. So of course the vesicles are very, very pure, but they are very, very small. So this does not really constitute an organelle at all. And then you can make phase diagrams. So here, since we basically have three parameters, budding rate, fusion rate, and maturation rate, we can normalize the budding rate and the maturation rate by the fusion rate, and that gives us 2 parameters. And this is very nice because we can represent them on phase diagram on the page. And so this is the budding rate over the maturation rate. This is over the fusion rate, sorry. This is the maturation rate over the fusion rate. And this is the purity of the compartment, the average purity. And purity is how far from the center point they are in this ternary phase diagram. So a system that is very A will be purity 1. The system that is mixed of 3 compartments will be purity 0. And as you can see, the purity, there is a purity transition which occurs when the budding rate is equal to the maturation rate. This is this line here. If the budding rate is smaller, then the system is mixed. If the budding rate is larger, then the system is pure. Regarding the size, so this is a typical size distribution we have. You can characterize an average size, and this is how the average size depends on the budding rate and maturation rate. And as you can see here, the size mostly depends on the budding rate, not so much on the maturation rate, a little bit. But basically, if you increase the budding rate, you decrease the size. If you decrease the budding rate, you increase the size, as we expect. And interestingly, if you want to have a system with large compartments, but that are well defined in identity, then you need to be somewhere in the middle. You need to be fairly precisely located here, which is interesting because one could argue, although it doesn't look at all the same in the term of spatial structure, one could argue that this is what the mammalian Golgi looks like, large compartments with well-defined identity. Okay, so we can explain this analytically. I won't get into the details. Now, of the different models of that have been made of the Golgi, one thing, there are basically 2 models that have been historically put in competition with one another. One is a model where the cisternae of the Golgi are stable, and that the cargo that goes through the Golgi is transported from one cisternae to the next by vesicles. And so in this case, the vesicles are going in an anterograde fashion from the entry to the exit, because this is what transports the cargo. The other model, so this is called Vesicular transport model. The other model is called the cisternal maturation or cisternal progression model. And in this case, the cargo remains in the cisternae. The cisternae evolve over time to go from a cis to a medial to a late to a trans identity. And the cargo does not need to leave the cisternae because the cisternae themselves are moving through the stack by changing identity. But all these enzymes that must remain in the Golgi, they must remain in the Golgi, so they must leave a cisternae that is moving up, and they must be moving back towards earlier cisternae by vesicular transport. So in this case, the vesicular transport is going in the retrograde direction. And you can look at this in this animation. So basically, the vesicular transport model, vesicles are going anterograde from entry to exit. The cisternal maturation model, vesicles are going in a retrograde fashion from exit to entry. And for those who are not at all familiar with organelles or the Golgi, you probably don't realize how fierce the debates have been between these 2 models for the last, I don't know, 50 years or something. People are really stronghold about which model is the most relevant for the Golgi, and they make experiments that prove their point, and they make experiments that disprove the other guy's point. And this has almost come to a point where maybe the defense of your own model is more important than really looking at what is actually happening, which is unfortunate. So with our model, we are not taking any stance because both processes are allowed in our model. Vesicles can bud, cisternal can undergo maturation. The way vesicles are directed is not through any sort of predefined rules, but are purely directed by the fact that a vesicle has an identity and it will fuse with a compartment that has a similar identity wherever this compartment is, whether it's a compartment that is later in the maturation process or earlier, we don't say. But what we can say is we can look in our simulation and look in which direction are vesicles going. Are they leaving early compartment to go to medial and late compartment, or are they leaving late compartment to go to medial and early compartments? And so basically we looked at this, and this is what we have. So this is, here we fix the maturation rate to a value that is almost equal to the fusion rate, so that we have as many blue, green, and red species in the system, and we change the budding rate. So as we change the budding rate, the purity of the compartments increases, because if you increase budding, you increase purity, and the size of the compartment decreases. And as you can see, you have a sigmoidal curve with a sort of purity transition when the budding rate is of the order the fusion rate. And this is what tells you about vesicular transport. If the budding rate is very small, vesicular transport is retrograde, meaning that it goes towards the bluer cisternae and it leaves the greener cisternae. I will not, I don't get into the details, but this is what it means. If the budding rate is very large, then vesicular transport is anterograde. It leaves the blue vesicles and it goes towards the greener, the blue compartment, sorry, and it goes towards the greener, the later compartments. Interestingly, in the middle, vesicular transport is actually centripetal. It's going towards the bluer, towards the medial Golgi, both from the cis Golgi and from the trans Golgi. And we can actually explain this also. So very quickly, I'll just tell you why this happens. If the budding rate is very small compared to the maturation rate, so it means that if you have a compartment, the compartment basically undergo maturation very fast. So if you have components that have not undergone maturation yet, when they bud, they will fuse with compartments that are less matured. On the other hand, if you bud vesicles, if you bud components that have undergone maturation, since this compartment itself is very much mature, this vesicle will fuse back with the same compartment, and so it will not generate vesicular transport. So vesicular transport is retrograde. On the other hand, if budding is very fast, this is exactly the same explanation but reversed. And in the case of to explain the centripetal direction of the vesicle, this is because if you have a compartment that is red, which means that it's a medial compartment, it has blue and green patches, so it can fuse with blue and the grid cisternae, so it tends to disappear. There's not so many compartments that are medial in this case, which means that basically the vesicles that will mostly create transport are those medial vesicles that fuse both from the cis and from the trans compartments to create new medial compartments. And basically this is what happens. So what we see here, which is actually very nice, I think, is the fact that the directionality of vesicular transport does not need to be predefined in advance. It spontaneously emerges together with the size and purity of the cisternae from individual rules that are purely based on specific budding and specific fusion of these vesicles. And so at the end of the day, and this is what I want to leave you with, we can make a phase diagram as we made before. Here we've got the budding rate divided by the fusion rate. Here we've got the maturation rate divided by the fusion rate. And we can split these diagrams into three very crude regions. These are not real phases, these are crude regions. One region which we call the mixed phase, where you've got large compartments that have a very indistinct composition because they are mixed. And this occurs when the budding rate is small or the maturation rate sufficiently large. Then on the other extreme, you've got the vesicular regime, the vesicular phase, where you basically have a bunch of vesicles that are very pure but very small. This is when the budding rate is very large, and whenever you fuse something together, they bud and they form vesicles again. And in between, you've got the sorted regime, where the compartments are large and pure because they get large enough because the budding rate is not that large, but they are pure because the budding rate is still larger than the maturation rate. And now I want to be provocative and say that if you compare the yeast of Golgi and the yeast of if you compare the Golgi of yeast and the Golgi of, let's say, mammalian cells, one possibility is that they are simply across the maturation, across the sorting regime, because, as I said, the yeast Golgi, the maturation rate is typically of the order of 1/1 minute, while in the mammalian Golgi, the maturation rate cannot be measured directly because these cisternae are actually at steady state, but the transport rate is an order of magnitude slower. So what we argue is that it is possible that the yeast Golgi is in the mixed state and that the mammalian Golgi is in the sorted state. And actually, what can be done is you can starve yeast by depriving it of glucose. And when you starve yeast, very interestingly, the yeast Golgi tend to stack up together. It doesn't form such a nice stack like this one, but it certainly forms a nicer stack, and the cisternae becomes more sorted. So it looks like, indeed, if you decrease the maturation rate, when you starve yeast, you can imagine that you slow down all enzymatic processes, including maturation. And so if you decrease the maturation rate, you could just be going through this transition. All right, so I have a lot of, I have a few slides about comparison with experiments, which I will not get into. I can get into it if you want during the questioning. But and I just want to acknowledge these two guys who did their PhD with me on this subject. Quentin Van, who's now a postdoc at Dresden, moving to Geneva soon. Jean-Patrick Vrel, who was a PhD with me, and he was at the same time a medical student. He was a bit crazy, he really liked to study, and now he's back at medical school. And I'm going to stop now, and I'm going to thank you for your attention, and hopefully we still have some time for questions.
Thank you, Pierre, for that excellent talk. Vijay, would you like to go ahead?
Yeah, so Pierre, fantastic talk. So if you go to the phase diagram where you sort of provocatively showed the thing with the yeast comparison.
This one?
No, here, for example. So in the model, you don't have any spatial structure, right? So there's just binding and binding fission. But in the yeast, I presume the so-called cisternae are also dispersed and moving around. So in the mixed case, there is a current in the model from the outermost regions to. So what does that mean in the case of yeast when the vesicles are all floating around?
Right. Okay. So yes, there is a current. So in my system, there is always a current because my system is somehow something like. Where is this sketch? Yeah, my system is something like this, right? So I assume that I have a source and a sink. And there is a current between the source and the sink. This is somehow by definition, because there is an influx coming from the source and an outflux, which could actually, the outflux could go back to the source, and it does, or go to the sink. It depends on basically some vesicles which are put in the systems can exit before they maturate, and then they exit through the source, and they can exit after maturation, then they exit through the sink. So you have a current by construction because you have these two boundaries. And the question is, what does the steady state look like in between? So if this is what the yeast Golgi look like, and of course, there is a current also in yeast, the cisternae themselves, they are diffusing around, but they will still originate from the endoplasmic reticulum, and they will still dissolve into vesicles that will go eventually to the plasma membrane. So there is a current. It's just that this current is not, in a sense, is not a spatial current that you can follow if you were just to look at a vesicle, because the vesicle diffuses. But if you look at, there is a net flux going from the ER to the plasma membrane in yeast, and of course there is another flux that is revert. Does this answer the
But
question?
I wonder, yeah, yes. But I also wonder, I mean, you said if you do some drug treatment, the Golgi system and sort of stack up, I mean, there is a spatial arrangement as there. There's a spatial patterning.
So, okay, so maybe I address this because I completely agree that I think, I mean, the first thing that fascinated me with the Golgi is the way it looks, the structure, and this is completely absent in my model. So one could say that this is very disappointing, and maybe it is. But already the model gives a lot of interesting things, and it's already quite complicated. So that's why I think it's worthwhile talking about it. Now, what is the role of this? So basically, one could ask the question, maybe the main difference between these two things is the spatial structure. Maybe there is a protein that allows this Golgi here to be stacked and not this one, because there are definitely proteins that glue cystine A together. And maybe this is what makes the difference. And one could definitely argue that this is the case. I mean, why would cystine A that are closer to one another end up being much slower in transporting would be a question, of course. But one could argue that the spatial structure is key. So maybe I can just tell you about one experiment, which does not show it isn't, but gives some interesting indication. It's an experiment where the spatial structure was completely modified. And here what these guys did, so it's a fairly recent paper from the Rothman lab, what these guys did was to glue the Golgi cystine A on mitochondria. So in red, you've got the mitochondrion, in white, you've got the Golgi cystine A. And as you can see, this is a mammalian cell where you would have a stacked otherwise. The stacked structure is completely messed up. Cystine A cannot move anymore. They are glued to mitochondria, but nevertheless, transport still occurs with the same sort of rates, a bit slowed down. It is slowed down a bit, but transport still occurs. And there is no obvious qualitative change. Everything is slowed down, but there's no obvious qualitative change. If we do that in our model, so in our model where cystine A can fuse with one another, we get this sort of vesicular transport pattern, retrograde, anterograde, and centripetal in the center. Now, if you prevent cystine A to fuse with one another, which means that you will prevent the medial cystine A from disappearing, then you get something like this. You have basically an abrupt transition from retrograde to anterograde transport as you change the budding rate. But you don't change this purity idea. You don't change the fact that basically you change very little about the description of the problem, except that in the transition zone you don't have this centripetal flux. Mm-hmm. So I want to use this to argue that the spatial structure is not key. It is interesting and important, but not key.
Nice, thanks.
Srilaja?
Yeah, very nice talk, and I'm very intrigued with this result that you can actually get Golgi cystine A to appear when organisms undergo nutrient starving. Now, what is interesting about that is in a modeling process such as the one you're building, how does one also incorporate partly Vijay was also asking the same, part of the question, like how does one incorporate the spatial organization? So budding yeast, for instance, in which presumably this experiment was done, undergo open mitosis. So do higher eukaryotic cells. So do you need like an organizing structure around which this cystine A can form when the vesicle sorting is occurring, but there is no 3D organization in terms of a Golgi? So for that 3D organization to appear, do you need an external force in addition to the vesicle sorting? And can the model help answer these kinds of questions as well?
Yeah, thank you. Okay, so the first thing I wanted just quickly to show, so this is something I didn't discuss, but basically, so these are the results that show that when you starve Cerevisiae, what you see happening, so Cerevisiae would be something like this. You would have cystine dispersed all across the cytoplasm. When you starve it, then what you see is the appearance of what is called Golgi mini stacks, which means that in the endoplasmic reticulum, you've got these endoplasmic reticulum exit sites, which are sites where there's a lot of vesicles coming out of the ER. And so this is basically a localized source of material for the Golgi, let's say.
Okay.
And in another form of yeast, which I forget the name of now, unfortunately, you see these mini stacks appearing at the ER exit sites. So here, obviously, one thing that is breaking the spatial symmetry is the fact that you have the source is punctual.
Right.
And so you have vesicle emitted from a particular location. And so these vesicles will mostly aggregate in that region. So one big question is, how do they manage to aggregate before diffusing away? I don't have the answer. Maybe there is some sort of, I don't know, slow, maybe diffusion is slow as soon as the system A becomes large enough, but they are kept in place, and you form these mini stacks. So I would say that one answer to your question is the fact that the influx is localized at these ER exit sites. Now, they also exist obviously in the mammalian Golgi. There are ER exit sites, but the Golgi in the mammalian, it looks like one big organelle, which is actually called a Golgi ribbon, as maybe you probably know. And this Golgi ribbon is entirely, the formation of this Golgi ribbon is completely dependent on microtubules. If you depolymerize microtubules, the Golgi ribbon disappears, and what you end up with is exactly that, Golgi mini stacks. So what I would say is that in the model, it would be quite, so we did some simulations with space in them, but we didn't, I mean, it's a big work, so we haven't finished. But the first thing to do would be to have space in the system and to have a localized influx. And this could break symmetry.
Right.
And this could lead to the formation of Golgi mini stacks. Now, if you want the full ribbon, then you have to put microtubules, you have to put microtubule organizing centers, because there's microtubule nucleators in the Golgi, and then it becomes a real mess, but very interesting.
Right. I was asking also from the point of, let's say, cell division. I probably should know this, but I don't know what happens when cells divide. The Golgi apparatus disassembles and reassembles.
It fragments. So it fragments, and I know it completely fragments, fragmentates, I guess. And then it reforms.
And it reforms. So I am wondering
if it doesn't disappear, but it gets fragmented.
Yeah. And then it might follow the yeast model, where it forms these localized small mini stacks and then sort of coalesces together into a full Golgi apparatus.
I think this is likely. I thought maybe I had a slide on this, but no, I don't have a slide on this. Yeah, it is likely that it does something like this, I would say. Yes, the mini stacks are probably the first level of self-organization, and they need to be there in order for the larger Golgi ribbon to form.
Okay. All right. Thank you.
Welcome.
Next would be Apratim.
Yeah, I just have a general comment and a question. So the comment, of course, is that compared to, say, Dr. Sharma's talk before this, so where biologists spend so much time looking at individual ARFs, individual RABs, individual snare proteins, and where, as a biologist, I find it remarkable that you can abstract from all of that and just come up with these models for, say, either endosome fusion or Golgi maturation. My question is more...
Maybe I just comment that, so we've got that thing published. I cannot tell you how difficult it was.
I'm sure.
A lot of biologist referees, and they did not share your excitement, not necessarily.
Yeah. And but the thing is, what I was wondering was, this is also, the model is also informed about the detail of the biology, right? So particularly talking of the Golgi, you said that these vesicles which are budding, they could fuse either with the source or with the sink, but overall they have a directionality because there is secretion. But now, as you look, probably you had a slide just on that, the Golgins, which are present either on the cis medial or trans Golgi, they also seem to be, if you relocalize a Golgi from a cis cisterna to the mitochondria, the cis vesicles would get to the mitochondria, right? So it is not really, there does seem to be other components which are directing this transportation direction, right? So say the specific Golgi proteins which are also doing some of this sorting, acting, probably like RAB effectors in effect, right?
Yeah, you're absolutely right. I'm just sorry, I'm organizing my slides. So this was supposed to be the last slide to tell you how many of the things were absent in this model. Maybe I should have put it here. So here there's space, right? The first thing is the fact that you've got a localized influx, the fact that you can have adhesion between cisternae, the fact that transport could be influenced by proximity, in particular because you've got these long tether molecules that can actually prevent vesicles to explore a larger volume. And yes, you're absolutely right that in my model I had 3 components, and because I didn't want more, because 2 was maybe a bit too simple and more was going to be too complicated. But of course, fusion is a highly complicated problem, a process that involves itself many molecules, and so this homotypic fusion or heterotypic fusion or specific fusion could be modulated by many things that are different from let's say molecules that are different from those that are affected by maturation. So in my model, maturation, budding, and fusion are all influenced by the same identity markers, and I have only 3 of them. But in fact, you could have such a larger complexity, and you can find a particular molecules that you target to the mitochondria, and then all the protein gets stuck there. This is definitely possible. And also, I mean, the last thing I want to say is that basically our goal was to do, okay, what is the simplest thing we can do that would show interesting features? But you can, as I said here, an unbiased model, but you can add some specificity at any point to get things that would be more robust. For instance, one thing that would robustly lead to anterograde transport would be the case where you would bud a vesicle and then the vesicle would undergo maturation before fusing. So obviously then this vesicle would not come back, it would fuse in an anterograde fashion. This occurs in our simulations, but only for very high maturation rates. You could change the code, or you could change the model so that it would occur more systematically, and then you would completely bias anterograde transport. So you're absolutely right to point out that there could be so many ways to make the thing more complex and more interesting.
Yeah, and the details we that do not know. Yes, yeah.
Sure, that's another reason not to put them in. Absolutely, yeah.
And the short circuit, is a prediction of the centripetal part of the phase diagram that will the medial Golgi then get engorged because vesicles are fusing from both directions, from cis and trans to the medial Golgi. Then you won't have a steady state size of the medial Golgi, right? The medial Golgi will keep growing, right, in the centripetal.
Right. So okay. So it depends what you mean by steady state. So you always have a steady state in the same sense that you have a steady state in, let's say, these time tracers, for instance. Here I've got a steady state size, but I've got a lot of fluctuations, right? Yeah. And same thing here. If I have this sort of sketch, okay, if I look at this compartment, it will disappear, but there will always be compartment there. So yes, there is some sort of a steady state, and it will influence the stochasticity of the system. And there are actually some drug treatments that have been reported. So these are in all my experimental slides that I didn't tell you about. But there's some drug treatments that have been reported to increase stochasticity. Where is this? Like the fact that cisternae are very transient is increased by mutating a fusion protein, for instance. So these are things that are not discussed very often in the biology literature because maybe, I mean, well, maybe because people didn't think it was interesting, but yes, this is very interesting. It is very interesting to not only discuss the steady state, but also discuss the fluctuation around it. This is very well understood in some fields of biology, but maybe not in others. And our model can address that.
Yeah. Thank you.
Thank you.
All right, Pierre, there are no more questions here. Thank you again for the fantastic talk. It was really interesting to see all the theory behind all your papers in firsthand. I'll pass on the baton to Vijay.
Yeah, thanks both Mehak and Pierre for the very nice talks, and also thanks Senthil for chairing the session.